most recent MDA meeting in regards to Duchenne muscular dystrophy and the more recent information about treatment options for patients with Duchenne, definitely gene therapy is the leading information and new exciting news about treatment options for patients with Duchenne. Also different RNA therapies and outcomes from the available RNA treatments that we have today. Some new exciting news about new therapies that may be coming down the pipeline. So those are all new exciting news in the field of Duchenne muscular dystrophy.
Yeah, so the primary messages that I took away there were actually a couple. I thought that there was some interesting information regarding exon skipping. I think that there was additional information on Adeleurin, and I think we were all interested in seeing some of the data coming out for gene therapy. And I think that it's nice to be able to see that there's additional data that supports the efficacy of some of the exon skipping drugs. There's additional information to suggest that there's efficacy of Adeleurin, and it's nice to be able to see some of the preliminary data that's come out on gene therapy. So gene therapy for Duchenne was also presented at this meeting. At this point, it's still relatively early in development. We are observing data coming from 3 different programs, 3 different companies. There's Solid Biosciences, there's Pfizer, as well as the Sarepta Therapeutics programs. All 3 of them were presenting data from their phase 1 or phase 1/2 studies. And what we had seen basically were updates. There were 3 separate presentations. The Solid Biosciences presentation showed updated data that they had on their original 6 patients. 3 were dosed at the lower dose and 3 were dosed at the higher dose. They were able to provide some efficacy measures looking at the North Star Ambulatory Assessment as well as ambulation, and comparing them to a large external control group and showing the different spaghetti plots compared to what was seen in the large external control group. And that was interesting to see. They also announced that they had dosed their 7th patient, and that the program, which was previously on hold, is now continuing. The Pfizer study also presented some data from their first 6 patients, and they also looked at some of the similar assessments. They specifically looked at the change in the North Star Ambulatory Assessment and looked at how that compared to the change in the North Star Ambulatory Assessment that was actually captured in the control group in that placebo arm from a previous clinical trial. And they showed that there was differences, that the patients were stabilized relative to what was seen in the external control, where patients were declining on the North Star Ambulatory Assessment. And finally, the presentation for Sarepta Therapeutics. This is the gene therapy study, once again delivering a miniaturized or microdystrophin similar to the prior 2 programs through AAV delivery. And they presented a double-blind placebo-controlled study of 41 patients. So these patients were randomized to either receive gene therapy or placebo in the first period of the study. And then they were presenting the data from that 1-year period in the first period of the study, comparing the North Star Ambulatory Assessment. And what was reported was that there was, if you look at the subgroups, they divided, these patients were ages 4 through 7, and they divided into 2 different subgroups, ages 4 and 5 and ages 6 and 7 at the time of enrollment. And if you looked at the patients who were in the 4 to 5 age group, you could see that the baseline characteristics of these 2 groups was fairly similar. And after 1 year, there was a separation that was seen in the North Star Ambulatory Assessment in the treated group versus the placebo group. In the older group, ages 6 and 7, there was a difference actually in the baseline North Star Ambulatory Assessment. And so it appeared that the patients who received placebo actually on average were a little bit milder and had higher North Star Ambulatory Assessments compared to the patients who received gene therapy. The separation was not as clean, and as a result, the overall analysis showed that there was some separation in the study, in the treatment versus the placebo group, but the separation did not reach statistical significance. Nonetheless, I believe the data is encouraging and is optimistic, I think, for the promise of gene therapy in general. And I think that overall, among these 3 programs, we are seeing positive signals that gene transfer of a miniaturized dystrophin, such as mini-dystrophin, microdystrophin, does hold some promise for the treatment for Duchenne muscular dystrophy.
So one of the presentations talked about an available or a study regarding a treatment option which includes RNA therapy for exon skipping for exon 45. This data is new and has recently become available, especially that the drug was just recently approved. So this kind of provides more information for clinicians and for researchers that they're looking at the safety profile for this treatment option, in addition to the level of the drug in the system and its bioavailability, and also the amount of dystrophin that was found, is reassuring and definitely promising.
So the Chasmersen data that was reported was through a phase 1 study. This was initially a double-blind, placebo-controlled dose escalation study, looking at the safety of and pharmacokinetics and pharmacodynamics of Chasmersen in patients who were amenable to exon 45 skipping. And what was observed was that this medication is generally safe and well tolerated, and there were really no serious adverse events that were observed in these patients. And they were able to detail the pharmacokinetics that were seen in this molecule. This is a PMO type molecule, and the pharmacokinetics was fairly similar to what was seen in other PMO molecules.
So I think what neurologists should consider when they have a patient with Duchenne muscular dystrophy is first and foremost try to identify the specific mutation, because now there are treatment options that are available for certain mutation subtypes. So unless we know the mutations and the different genetic defects, then we may not be able to offer these treatment options. So this is very essential. And also considering that this may be a motive to encourage newborn screening in the future, since this may shorten the time to diagnosis and may help us offer these treatment options earlier, and may help even provide a better outcome for patients.
So there was a poster that was available for review that talked about Alexion. I think the patients were an open label extension, were enrolled in an open label extension study that they called Study 19. And it was, patients had been on Alexion for some time, and they looked at this cohort of patients and compared them to an external control, patients that had been followed in a separate study through the Synergy network. And given that there were reasonably comparable standards of care that were applied to these two groups, they looked at the time to loss of ambulation in these two groups to see if there were any differences. The patients in the Synergy network, on average, were losing ambulation at about 13 years of age. And that was compared to the patients that were in the in the study 19, those are the patients who had been on the open label extension study for Adelauren, and these patients on average were showing a loss of ambulation of greater than 15 years of age. And so this difference was nice to see, because we've been observing the clinical trials for Adelauren for quite some time, and it's has conditional approval in the European Union, but it does not yet have approval in the United States, and we're wondering what the FDA decision will be in the end for Adelauren.
Some of the new information that was provided in the meeting in regards to RNA therapies is first and foremost is that they are safe and well tolerated in patients who receive them. And there was the exciting news is that in one of the presentations that there was a delay in loss of ambulation by at least 2 years in regards to compared to patients who have received the standard of care such as steroid therapy. So there is benefit from the prolongation of the duration of ambulation and delay in the loss of ambulation time, which is significant. So I think this is all exciting because it's offering options for patients to choose from, and also proving that these treatment options are so far safe and well tolerated by most patients.
And finally, there was some data that was presented by Craig McDonald on Teplersen. Teplersen is the PMO molecule that's designed to skip exon 51. This is a medication that was approved now almost 5 years ago, 4.5 years ago, and is being used commercially. And so what was done was these patients that were studied in various different studies for Teplersen were pooled together. And many of these patients have been on Teplersen for quite some time, open label, through an early and young patients, as well as non-ambulatory patients and ambulatory patients, and pulling all these patients together and comparing them to an external control group through the Synergy Natural History study. And so when they were pooling these patients, they looked at various different measures, but most importantly, loss of ambulation. And what was seen among all the patients when they were pulled together was that the average age of loss of ambulation was just over 15 years of age, and that deviates from what is seen in the natural history external control group. There were a couple of different control groups that were used. They specifically isolated patients who were genotypically similar, so patients who had a DNA testing that suggested they were amenable to exon 51 skipping, but were not receiving Teplersen. And what was seen there was a significant difference. These patients on average were losing ambulation at approximately 13 years of age. There was another external control group that was also compared to this patients who were not just exon 51 amenable skipping, but a larger group of patients in the Synergy Natural History study. And those patients were closer to about 14 years of age at the time of loss of ambulation. But this was used as a sensitivity analysis to show if the conclusions of comparing to the exon 51 amenable patients was a reasonable conclusion. And they reached a similar conclusion when they broadened the control group to include more DMD patients that were followed in the Synergy Natural History, which is that there is a significant difference in the time to loss of ambulation when you control patients who are on Teplersen compared to patients who were not treated with this exon skipping medication. I think this is interesting. I think that this provides us additional confidence that there is some disease-modifying activity of Teplersen in patients who are amenable to exon 51 skipping. This is a medication which is commercially available, and it's helpful for us as practicing physicians taking care of DMD patients because it provides us additional data and confidence that we're providing a medication which is efficacious.
In regards to the gene therapy platform for limb-girdle muscular dystrophies, I know there are several works that's being done. In terms of the information presented in the Muscular Dystrophy Association meeting, there was data on limb-girdle muscular dystrophy type 2E, and it has shown promising results, including the efficacy and the safety of the gene therapy in these patients with limb-girdle muscular dystrophy type 2E. I think it would be interesting and curious to know long-term results as well as similar trials in other types of limb-girdle muscular dystrophies. And there was one-time intravenous dosing of this gene therapy product, and that has shown improvements in the functional outcome, including some outcome measures that we commonly use in the clinical practice, such as North Star ambulatory measurements and other functional assessments. I think it is critical that we have that information right now, which can help us with more research towards similar gene therapy trials for other limb-girdle muscular dystrophies, and also treating these patients with limb-girdle muscular dystrophy type 2E in the future.
In addition to Duchenne, there was presentations regarding limb-girdle muscular dystrophy, which we know that this is, until recently, there's a big unmet need for patients with limb-girdle muscular dystrophy to be offered treatment options. And some are more common than others. So one of the presentations talked about, again, the delivery through a viral vector for gene therapy for one of the common or more common types of limb-girdle muscular dystrophy. So once a vector is identified, I think this will open the door for many different types of limb-girdle muscular dystrophy, other dystrophies, to hopefully be offered gene therapies. Of course, it will still have to be tailored to each and every type of genetic defect and each and every different type of the dystrophy itself. But I think this was exciting because this is new, and it will provide treatment options for another subtype of patients with neuromuscular disease.
In terms of new clinical information pertaining to limb-girdle muscular dystrophy that was presented in this conference, I was curious, and it was interesting for me to look at some of the information that was presented from advocacy organizations and patient support groups, that even now there is delay in the diagnosis of limb-girdle muscular dystrophy in the community. I think that tells us about the education and awareness we still need to recognize and diagnose these conditions. We need more providers, including primary care providers, pediatricians, to be more aware of these limb-girdle muscular dystrophies for prompt referral so that they can be diagnosed as soon as they can, and then they can get onto the treatment or other management.
So one of the challenges of doing clinical trials in limb-girdle muscular dystrophy is that this is a rarer disease than Duchenne muscular dystrophy. So it's harder to recruit the numbers of patients that would give us the statistical power to be able to show clinical efficacy. Nonetheless, something as powerful as gene therapy certainly could potentially provide efficacy that could be captured even in smaller numbers. And I think that that's kind of what they're hoping for. There are additional limb-girdle muscular dystrophies besides the ones that are currently being studied. The one that's currently being studied is limb-girdle muscular dystrophy 2E, which are mutations in beta-sarcoglycan. There are other limb-girdle muscular dystrophies besides 2E that sometimes tends to be milder, and that presents an additional challenge because milder patients tend to be have more variability in their phenotype, so their variability in their severity. So that's the time when you're going to need larger numbers of patients to be enrolled in the clinical trials to be able to show efficacy. So I think that although this is a promising next step, the next muscular dystrophy perhaps next set of muscular dystrophies that perhaps may be amenable to treatment, especially with something as powerful as gene therapy, I think we immediately see some challenges in terms of what the clinical researchers are going to be able to accomplish. And I hope that we're going to be able to overcome these challenges so that we can have some approved treatments for these other patients.